Hello, everyone. It's a pleasure to be here virtually today, and I appreciate the invitation. I was asked to talk about the basic science of Dravet syndrome. So, I'm Ethan Goldberg. I'm an assistant professor and pediatric neurologist at the Children's Hospital of Philadelphia in Philadelphia, Pennsylvania, and I run a basic biomedical research lab studying basic mechanisms of seizures and epilepsy. And I'm also an attending in our epilepsy neurogenetics program, and I serve on the Dravet Syndrome Foundation Scientific Advisory Board. So I have no disclosures today. I will be talking about some background about basic science, about the basic science of Dravet syndrome, and a little bit about some of the work ongoing in my laboratory on the subject. To start, I wanted to tell you very briefly about our epilepsy neurogenetics program. Program is a little over a year old, and the goal of the program is to integrate the results of genetic testing into the care of children with epilepsy, as well as to enhance access to genetic testing, enroll patients in clinical research studies, and then on the basic science side, to use the results of genetic testing to drive the development of new treatments. Here's a little overview of our program. So we have an intake core where patients who come into our program, either from Philadelphia, from the Mid-Atlantic area, anywhere in the country, and even through our global patient services program anywhere in the world, evaluated for potential genetic testing, as well as evaluation by various members of our multidisciplinary care team, including physical and occupational therapists, experts in neurogenetic care and testing, neurogenetic counseling, EEG. We have a very large ketogenic diet program, and collaborators in other subspecialties important for the care of children with complex care needs like cardiology, gastroenterology, neuro-ophthalmology, et cetera. And we've expanded where we now have 10 attending neurologists with subspecialty expertise in neurogenetics, 5 neurogenetic counselors and therapists embedded in our program, where we offer access to advanced genetic testing, enrollment in clinical research studies, and access to advanced clinical trials in the field. So we, in the first year of our program, so this is FY20, so we're just getting some data from this year, but we did over 750 neurogenetic evaluations for epilepsy in FY20, and we're going to exceed 1,000 this year. One of the other goals of our program is to make sure that every patient gets seen by an attending physician with subdomain expertise in epilepsy neurogenetics, as well as a neurogenetic counselor with expertise in epilepsy neurogenetics, and we increased the proportion of patients who had access to genetic counseling from 28% to 85%, and that's now over 90%. And we were able to offer over 90%, it's actually now over 95% of all patients access to advanced genetic testing while cutting down on wait times, as well as median time to diagnosis, you can see now is under 2 months. And in the age of COVID-19, we expanded a telemedicine program where we delivered epilepsy neurogenetics care to children and their families in 25 states around the country, as well as 9 countries outside the United States, including on 4 continents. And this is just some data on what we're finding. Of course, our number 1 diagnosis is Dravet syndrome or Dravet syndrome-related disorders, where we identify pathogenic variants in the gene SCN1A, encoding the voltage-gated sodium channel type 1A, NAV1.1. So I wanted to give just a brief intro on the basics of basic science in Dravet syndrome. So of course, we start with a gene, and that gene is SCN1A, again, which encodes the alpha subunit, which is the pore-forming subunit through which sodium flows. And this is a channel complex that's made up of multiple proteins. And this channel is expressed by neurons. There are different types of neurons. So here we have an excitatory cell. It releases glutamate. And its activity is regulated by different subsets of inhibitory cells that release the neurotransmitter GABA. So this cell is marked by a substance called PV or parvalbumin. Its synapses are around the cell body of this excitatory cell. It regulates action potential generation. This inhibitory interneuron, this red one, is marked by another peptide called somatostatin. Its axon goes up to the top of the brain where it impacts on the dendrites, which is the receiving end of the excitatory cell. So it regulates synaptic integration. And then the third major subclass of interneuron expresses VIP, vasoactive intestinal peptide. it inhibits other interneurons. So it's like a double negative. It's disinhibitory. When it inhibits the somatostatin cells, it relieves inhibition on the distal dendrites of the pyramidal cell and thereby promotes excitation. So these circuits, we think, underlie complex behavior, such as memory formation, social interaction, as well as underlying whole animal or whole individual manifestations of disease like seizures, developmental delay or intellectual disability, and to phenotypes of autism and SUDEP. So I think one can divide the basic science of Dravet syndrome sort of along the lines of what I just discussed. So there are basic questions about what gene is involved. SCN1A, how is its expression regulated? So we're missing one copy of the gene. Why is it that the body can't just make more from the other gene? Are there other genes that regulate its expression? Other elements of our genome that regulate its expression? How do genetic variants in SCN1A alter expression of the protein? Then as we move from the genetic level to the ion channel level, how do these variants that we identify in SCN1A affect how that channel functions? And then we march down the line. So we can then say, how does that altered ion channel function impact how a neuron operates? Which neurons are important? Is it all of the neurons? In the cerebral cortex, which we think of as the seizure generating part of the brain, there may be over 100 different neuron types. And those neurons are different from each other for various reasons by their anatomy, what neurotransmitter they release, but also what ion channels they express. So how does absence or presence of SCN1A in a particular neuron regulate its activity, and how is that impacted by a pathogenic variant in SCN1A? So now when we go to circuits, we can look at intact circuits in experimental animal models, for example, in brain slices. We can make a computational model and manipulate its activity, and we can see how does altering the activity of defined subsets of neurons alter circuit activity, synaptic transmission, communication between neurons, rhythmogenesis, so the rhythmic activity of the brain that's important for attention, for memory formation, et cetera, as well as for seizure generation. And then we can look at a behaving experimental animal, whether that's a mouse, a fly, a fish, et cetera, that's missing one copy of SCN1A to study whole animal features like seizure generation, SUDEP behavior, et cetera, okay? So why should we care about this? There's a big push, I think, towards translational research. And I think when we have a syndrome where children and their families are suffering, there's enormous pressure to try to put our resources into or all of our efforts towards a cure. But it's important to understand that some of the exciting advances that have been made recently stand on the shoulders of basic science research. So just as an example, there are innumerable prior scientific discoveries that were built upon basic science research. For example, the internet or the invention of a laser. Other key advances in medicine and healthcare, like treatment for Parkinson's disease, medication to control cholesterol, which reduces risk for heart attack, for stroke, as well as cancer chemotherapy. So basic science leads to an understanding of the cause of disease. So we would not have a Dravet syndrome foundation. We would not have a diagnosis if we did not know about sodium channel genes. We did not know about SCN1A. We did not know that it was genetic variants in SCN1A that led to Dravet syndrome, as well as basic science discoveries as to how genetic variation in SCN1A altered the function of ion channels. So we know, for example, that it is due to so-called loss of function. So those genetic variants impair the function of the ion channel and have led to the so-called interneuron hypothesis. So I mentioned about these different types of neurons. SCN1A is not homogeneously expressed across all of them. It's relatively selectively expressed or preferentially important for the function of these interneurons. Less sodium channels in interneurons impairs the excitability of those cells. There's less inhibition, which leads to it's an oversimplification, but an imbalance between synaptic excitation and inhibition, which biases the system towards seizures. So this basic science understanding allowed us to develop models that we can use for high throughput drug screening, for testing gene-based therapies like antisense oligonucleotides and gene therapy. And there are models that have been developed in mice, in flies, as well as in zebrafish, that very closely recapitulate the core features of Dravet syndrome. And these have been critical towards understanding the pathophysiology of various features of Dravet syndrome, like how seizures are generated, what is the mechanism of SUDEP, et cetera. And some of the new drugs that we have from Stoke Therapeutics, for example, and other emerging new therapies were critically dependent upon the availability of these validated experimental animal models. And I think a key point here is that support for basic science keeps science in the public interest. And what I mean by that is our basic biomedical research system in the United States, which is funded through grants, that are through the government, that are through private foundations like the Dravet Syndrome Foundation, that are geared towards what the public wants us to be studying. Okay, so here's just a graph. This is showing PubMed citations. So these are papers about Dravet syndrome, and we can see since the discovery of the genetic cause of Dravet syndrome, there's been an explosion in scientific activity on Dravet syndrome, and there in approximately $15 million this year in federal funding towards basic science research studying Dravet syndrome, mainly from NIH, most of which is through NINDS, the National Institute of Neurological Disease and Stroke. So there are 34 funded NIH research grants ongoing studying Dravet syndrome as well. And there's also funding from various private funders, including Dravet Syndrome Foundation, Dravet Syndrome UK, et cetera. Okay, so this is data, so-called RNA-seq data from a public database. And what this shows is the different brain-expressed sodium channels. So here's SCN1A. There are 9 sodium channels, 4 of which are expressed in the brain. And we can see on the x-axis age. And what we can notice here from this data, we're looking at RNA transcripts from single neurons, and then we can count them up. And we can see that early on, SCN3A is expressed at high levels. And then the expression of 3A goes down, and we see upregulation of SCN1A. And that makes sense because the mean age of first seizure in Dravet syndrome is at about 5 months, right? When we see this switch from SCN3A to SCN1A. So this data can make predictions about when we're going to see disease onset, okay? Another approach at the ion channel level is to do recordings of sodium channels that harbor Dravet syndrome-related dysfunction. So in this case, we have some cells in a dish, right? So these are HEK cells. These are mammalian skin cells, but they don't express ion channels. So now we can add genes for SCN1A, either wild type or variant, along with some of the other friends that SCN1A hangs out with, these beta subunits. And then we can stick an electrode in these cells and we can record the normal sodium channel versus the sodium channel harboring this variant. So in this particular example, we can hold the neuron. Again, the cell, in this case, we're studying a skin cell. It's like a little battery. So we can hold the cell at a negative membrane potential, which is like the inside of a neuron. It's negative on the inside relative to the outside. And then we can increase the voltage to different levels, and that's going to activate the sodium channel. So in this case, we see these different lines. This is called a family of currents. So we can see this light pink line, which produces this deflection, and then the dark pink line when we go all the way from -90 to 0. And by convention, the sodium current is inward. So one thing we can notice is this rapid time scale. So the channel opens and closes very fast on the order of milliseconds, and it's activated by voltage. And sodium, positive current, and by this convention is downward, flows into the cell. Okay, and then we can make this little chart plotting the amplitude of the depolarization against the amount of current that we get. And we can study different features of this current, how much current goes through, how fast does the channel close, etc. And here's an early paper showing this, where this particular group studied the wild type or the typical sodium channel, and we can see that that current is, in this case, 4, it doesn't matter what the unit is, but in this case, it's nanoamps, 4 nanoamps, which is a big current, whereas most of these other currents are very, very tall, so very, very small. So you can see there's almost no current going through this particular variant, this G979R variant, and all of these show what's called loss of function. So there's less current. And that's because either the channel is broken, it doesn't conduct sodium, or it doesn't move to the cell membrane where it needs to be. Okay, here's an example where, in this case, we can take a patient's cells and we can, in this case, blood or skin, and we can reprogram it into a pluripotent stem cell, which we can then we have a stem cell line from a patient with Dravet syndrome that we can edit using CRISPR to correct the Dravet syndrome mutation. And then we can turn those into neurons that either are human neurons from the patient or are isogenic, so same genetic background, but lacking the Dravet causing mutation, and then see how that SCN1A variant affects the activity of the neuron. In this case, we can see these beautiful neurons in the dish that we can target for electrical recording. Okay. So we were interested in this idea, and we were particularly interested in how the features of Dravet syndrome interacted with development. So we took a Dravet syndrome mouse, here's an SCN1A mouse, and we crossed it with a mouse line where we could mark inhibitory interneurons that were labeled red. And we could then record those interneurons at different points in development. Most of the work in Dravet syndrome had been in the first 2 to 3 postnatal weeks of life in the mouse, which is like an infant human being. It's impossible to sort of compare directly because mice and humans are very different, but there are a lot of similarities that allow us to model Dravet in the mouse. We can then make brain slices. So here's a picture of a 300 micron thick brain slice. We're under high magnification. We can target these inhibitory brain cells for electrical recording. And this is what we found. At postnatal day 20, we can see that the wild type neuron, in this case, we're injecting current into this cell, and this cell is discharging action potentials, which are electrical discharges that are the unitary currency of information transfer in the brain. And this cell, this parvalbumin interneuron, or so-called high frequency firing interneuron, because it fires so fast, discharges these action currents at over 400 per second. Whereas the Dravet syndrome inhibitory interneuron, you can see the amplitude of these little line slowly goes down until the cell fails to generate action potentials, okay? So this has been well established in the literature. What we found is if we waited a few weeks until the sort of juvenile period in the mouse, these fast spiking interneurons had completely corrected themselves, which was curious to us. So that led to 2 important questions. So we know in the Dravet syndrome mouse, as this process is ongoing from early juvenile to young adult mice, about half the mice are succumbing to early death from SUDEP or status epilepticus. So since we can only record from these surviving mice, from the mice that have survived to this point, how do we know that the interneurons in these cells were ever abnormal? So were abnormal and switched, or were they always like this in the cells that in the mice that had survived? The second question is, these mice are not normal. They continue to have features of Dravet syndrome, epilepsy, SUDEP, endophenotypes of autism. So if the interneurons have corrected themselves, why do we still see features of Dravet syndrome? So to address this, Keisuke Kaneko, who's a postdoctoral fellow in my lab and has now transitioned to independence as an assistant professor in Tokyo, he generated a procedure called mini slices, where he performed a microbiopsy from the cerebral cortex of mice at a young age, in this case between postnatal days 16 and 21, so an infant mouse. And then he did electrical recordings from the interneurons, and he did this from multiple cells simultaneously. A subset of those mice are deceased from SUDEP. Of those that survive, though, and those in this case, in this nomenclature, are group C. So the mice that survive are group B at young age, and then at this later time point, he then made conventional brain slices, so he could record from the interneurons at both young and late age from the same mice. And then we could compare to wild type mice that had 2 normal copies of SCN1A at both ages. And he confirmed that in both group A and group B, so this is the SCN1A mice at young age that subsequently died, as well as the SCN1A mice at young age that survived, all of the PV interneurons were abnormal. None of them could sustain high frequency firing. Whereas we reconfirmed in group C, these are the SCN1A mice that survived, recorded at older ages, they were just like wild type. Okay, so this shows that all of the PV interneurons their spike generation is abnormal, And of those that survive, their spike generation corrects, okay? So he then recorded simultaneously from these inhibitory interneurons here that we mark fluorescent red. So here you can see a brain slice. There's lots of cells here. The inhibitory interneurons are marked red. And he recorded from these inhibitory interneurons here, as well as the target excitatory cells. So now he can record not just the activity of the cells, but their connections to other cells. And he then stimulated the inhibitory cell at various frequencies and recorded the output measure, the synaptic activity, which is these little blips here, these little deflections in the postsynaptic excitatory cell. And what he found is that the message generated in cell 1 in the PV cell didn't make it to the postsynaptic cell about a third of the time. So we see that there's a failure of action potential propagation. Synaptic transmission is failing in a frequency dependent manner. So as we start stimulating faster and faster, these red arrowheads are indicating failure of synaptic transmission from the inhibitory interneuron to the postsynaptic excitatory cell. We can also look at the latency. So the time from that action potential in the inhibitory cell to the postsynaptic event recorded in the pyramidal cell is prolonged. So inhibitory synaptic transmission and its activity depends critically on timing. So we can see that that timing is disrupted in Dravet mice as well. So that was a brief overview, but just to summarize, we found that the impairment of interneuron spike generation in Dravet syndrome is actually transient. It exists in a narrow developmental time window. But we found that this impairment in synaptic transmission, which is sort of an indirect measure of action potential propagation, is long lasting, okay? So what we think is that this combined early impairment in spike generation and propagation may correlate with disease severity. So those mice that are deceased from SUDEP, we find that there is impairment both in spike generation and in synaptic transmission, and we're developing this as potentially as a prediction tool, again, in an experimental model system for disease severity in SUDEP. So very briefly, I wanted to turn back to this circuit diagram. I sort of mentioned these VIP interneurons as these kind of mysterious cells. We know that these cells are important for how the brain functions. They're disinhibitory in cerebral microcircuits. They receive ascending acetylcholinergic modulation, so they have a lot of acetylcholine receptors. And what's thought is that ascending cholinergic modulation activates these cells, which then inhibits somatostatin cells, which relieves distal dendritic inhibition and wakes up the cerebral cortex. So VIP cells are part of the mechanism whereby acetylcholine increases attention. And we thought that VIP interneuron dysfunction could relate to some of the non-seizure comorbidities that we see in Dravet syndrome. And in fact, this is RNA-seq data from the Allen Brain Atlas. We can see, so here are PV cells. About 60% of the SCN, the sodium channel transcript is SCN1A. But for VIP cells, they also express SCN1A, not as much as those parvalbumin cells, but they express quite a bit. So Kevin, who is an MD PhD student in my laboratory, he took the same basic approach and recorded from these labeled VIP cells, and he found that they, I'm glossing over some of the details here, but a subset of them was prominently impaired. And in fact, as opposed to the parvalbumin cells, they remained impaired. So even in the young adult mice, they were very impaired. So this is a input-output curve. As we inject more current, we count the spikes that come out the other end. Here's wild type, and we can see that the mutant is very impaired. So Kevin then went on, pardon me here while I try to play this video. He then imaged the activity of these VIP cells in a living animal. So we can get an output measure of cholinergic tone because it correlates with pupil dilatation in this head-fixed mouse. So this is a Dravet mouse, and we can use a deep learning algorithm called Deep Lab Cut, which we can train to monitor the size of the pupil, which is again an indirect output measure of brain state. And we can see that as this animal is This is locomotion here. So this white trace here is as the animal is running on a spherical treadmill, its head fixed. And we're imaging with a technique called two-photon microscopy. This is just raw data, but you can see that the green cells are the excitatory cells, and these red cells here, the tiny red dots, are the VIP cells. And what you can see is that the VIP cells, once the animal starts running, are activated, and that leads to activation of the excitatory cells. So as the animal starts running, you can see that many, many more cells become active. And then we can quantify that. And the advantage of the two-photon microscopy is that we're using an indirect microscopic measure of activity. So these cells are increase their fluorescence when they're firing action potentials. And that allows us to record the activity of hundreds or even thousands of cells simultaneously, whereas with the electrical recordings, we could only record 2 or 3 or at the most 8 cells. So what this shows here, let me get my laser pointer back, pardon me. Here's the VIP cells in wild type. Again, you can see pupillometry here at the top, and then this is running bouts here at the bottom. And you can see the VIP cells are in lockstep with transition from quiet wakefulness to locomotion, which also correlates with pupillary dilatation. And that then in turn leads to activation of the excitatory cells. Where in the Dravet mice, that's impaired. Okay, so now you see that only half of the VIP cells are paying attention, and that leads to impaired brain state transitioning. So the cerebral cortex is not appropriately waking up in response to ascending cholinergic tone from the brainstem. So to summarize this last part of my talk, what we showed is that these VIP interneurons, and I glossed over many of the details, but the papers are published, also expressed NAV 1.1. So it's not just these fast spiking interneurons. These VIP cells have this sort of counterintuitive role, we think, in the pathomechanisms of Dravet syndrome, likely the non-seizure manifestations, attention, memory, brain state transition, et cetera. And we found that this dysfunction, in contrast to the parvalbumin interneuron dysfunction, is actually long-lasting. It persists. We showed that this brain state transition is impaired in the SCN1A mice in vivo, in intact animals in vivo. And what we think is that manipulating or targeting these VIP interneurons may be a means to target some of the non-seizure manifestations of Dravet syndrome. So just to conclude my talk, I appreciate your attention. I still think even for the most common genetic epilepsy, SCN1A related disorders, we still have an incomplete understanding about how genetic variation leads to disease, but we've made an enormous amount of progress. The mechanisms of different endophenotypes of this complex disorder may be different. So the mechanism of seizure generation, for example, may be quite different than the mechanism of forms of cognitive impairment or of SUDEP. What I also showed is that the so-called mechanism, whether it's at the level of a cell or circuit, may actually be transient. And what I mean by that is that when we examine the situation at one point in time, we may find that a different cell or cells or circuit manifestation is abnormal at a later time. And this suggests that there may be different windows for intervention depending upon what the target is. And that leads to questions about which cell types or which brain areas should be targeted for manipulation and when should that occur. And leads to this concept that we've developed where the mechanism of disease may be a so-called moving target, where the actual target for a particular intervention may be different at different points in time. So with that, I will conclude, and I would like to thank members of my lab. In bold are individuals who are directly working on Dravet syndrome, and I mentioned the work of Kevin Goff, again, who's an MD PhD student in my lab, Joanna Mattis, who is an epilepsy fellow at the University of Pennsylvania, MD PhD adult epileptologist who's been studying Dravet syndrome. Laura McGarry is a child neurology trainee in my laboratory. Ala Somarathu is a Dravet Syndrome Foundation funded postdoctoral fellow. Nate Satuyo in my lab is a graduate student who's co-mentored by myself, as well as my collaborator, Stuart Anderson, and he is using cell transplantation as a potential treatment for Dravet syndrome, as well as former members of my lab, Morgana Favreau, who did the developmental parvalbumin interneuron story, Connie Tran, who's now a neurology resident at UT Southwestern, and she did some of our imaging studies in Dravet syndrome, and then Keisuke Kaneko, who's a former postdoctoral fellow in my lab who did the multiple whole cell recording. And we collaborate widely with many laboratories. I would like to acknowledge many of our collaborators, as well as my coworkers in the Epilepsy Neurogenetics Initiative, in particular, Catherine and Ingo Helbig and the rest of our ENGIN team, as well as our funding sources, most prominently from the Children's Hospital of Philadelphia. The Dana Foundation funded some of our neuroimaging studies, the Dravet Syndrome Foundation, of course, for generous support, as well as the National Institute of Health. And with that, I will conclude. And I've included here our ENGIN email address. If there are questions about the content of this talk, or there are questions about clinical care, etc., please feel free to contact us. Thank you very much.